Recently in lab, we've been talking a great deal about the plasmid and the way in which certain genes on that plasmid are expressed based upon certain environmental conditions. But the hard thing is that in lecture we really haven't yet covered the way in which DNA on that plasmid is converted to RNA and then that RNA is converted into protein. So let's turn our attention in this lecture to gene expression and the central dogma of gene transfer. It's going to be a wild ride, so hold on tight. Let's begin by looking at the bacterial genome and noting that the genome of a bacterial cell is technically defined as the chromosome and the plasmid. People get really lazy about this, and in fact, oftentimes you'll see that they'll refer interchangeably to the chromosome and the genome. In fact, referring to the chromosome as the genome of a bacterial cell. But in fact, in order to be entirely accurate, we would want to incorporate the plasmids into that broader definition of what the genome of the cell is. Because despite the fact that plasmids are not required for the survival of the cell, as we know, they can give it many incredible traits that may add to its superhero powers in many, many ways. So, in any time that we talk about the bacterial genome or that we talk about DNA, the term that always comes up is the gene. And it's interesting that for a term that is so widely and broadly used, we really don't have a fantastic definition of a gene. Suffice it to say that a gene is generally something that encodes for a specific product. Now, that product, you'll notice I put in parentheses there, is often a protein. And you might be thinking to yourself, well, isn't it always a protein? But in fact, if you think just a little bit more, you'll remember that in fact sometimes a gene might encode for something that simply stays as an RNA. I can't remember who it was, but somebody on their exam wrote that wrote about the tRNA and the rRNA and their roles in gene expression. And in fact, they play an incredibly important, inordinately important role in gene expression as RNAs. So the tRNA and the rRNA, they're never converted into protein, but they do their jobs as RNAs. So with that in mind, let's take a little bit more time to look at the central dogma of gene transfer, the way in which DNA encodes for RNA, which then maybe encodes for a protein, maybe or not, depending upon what the product is, the one specific product that gene encodes for. This picture is the epic central dogma of gene transfer, and it is something that perhaps you have seen in another class and that you will definitely see again. So the central dogma explains the way in which DNA gets duplicated, the way in which DNA gets converted into RNA, whether that be mRNA or not, and the way in which mRNA gets converted into the protein product. Let's start with the process by which the DNA gets duplicated, and it doesn't matter whether we're talking here having this circular piece of DNA represent a plasmid or whether it's representing the circular chromosome of a cell. Either way, we know that it has its origin of replication and can be duplicated. That process is called, fittingly, replication. This is actually where we will begin our coverage, and we'll talk about the real nuances of DNA replication. We'll also be looking at the process by which DNA code gets read and converted into RNA. And you can see in this process an awesome enzyme called RNA polymerase reading along the DNA and converting that code into a temporary encrypted message in the form of RNA, and particularly mRNA. So some of you might recall that this process of converting DNA into RNA is given a name. It is called transcription. So this decryption of RNA into a temporary, or decryption of DNA into a temporary RNA code is very important and referred to as transcription. Now, we recognize that the RNA that results from transcription, it could potentially just stay as RNA. rRNAs that are a part of the ribosome or tRNAs that deliver amino acids to that ribosome, they stay as RNAs. However, mRNA is the one that goes on, and mRNAs do encode for eventual protein product. So we can recognize mRNA as messenger RNA, carrying temporarily the encrypted code from the DNA to allow for the building of the protein product. So the ribosome now reads that mRNA, scanning down the mRNA and reading a little at a time and converting that code into polypeptide chain. And we see that polypeptide chain sticking its head out of the large subunit of the ribosome and getting created there. Of course, we now know that that protein will fold into its secondary, tertiary, and quaternary structures, perhaps, and take on its overall structure and function. This process by which RNA, mRNA, is converted into protein product is called translation. So, transcription and translation, two very different processes. Transcription converting DNA to RNA, translation converting RNA into polypeptide code. So this is a really important set of processes, and maybe right now you're thinking to yourselves, yeah, Rachel, I've heard it all before. I had this in kindergarten. Maybe you're thinking that if you're a prodigy, perhaps. But in fact, these become very nuanced. And after a while, when I start throwing out terms like things like TUS, terminator utilization substance, and the promoter, and things like primosome, there'll be a lot of terms that'll come up that are nuanced parts of transcription or translation, and then you'll be like, wow, I'm not sure I quite understood this as well as I thought I did. So that's why I like to start with the big picture. So keep this big picture in mind and keep coming back, flipping back in your notes to this big picture so that you're able to keep the big picture in mind through all of the little details. That is, we want to be able to see the forest through the trees. We'll begin with the process of replication, the duplication of either a plasmid or a chromosome. No matter the case, we still start at an origin of replication. For our purposes, we're going to look at the E. coli chromosome. We're using that as an example because, quite simply, it is the most well typified. That doesn't mean that it's actually simple, however. It's quite a complex process. There's about 30 different enzymes, protein complexes involved in this process. So even a tiny chromosome relatively, like E. coli, has a lot of nuances in the process. But in any case, it begins at what we call the origin of replication. This origin provides, if you will, the landing pad for all of the complexes involved in replication. And in fact, two replisomes, which with their complexes to build new DNA, will bind at this site. You can see here, and let me actually try to sort of show you what you're seeing here. This is one replisome right there, and this is another, so I've just circled the two. And as they do their job in making new DNA, copying the DNA, this replisome here will head this direction, reading the existing DNA as code and making new DNA. This replisome will head the opposite direction, again, building new DNA. You can see how eventually over at the other side they're going to meet. And they're going to meet at a place called the termination site. And in fact, there is a special protein that binds at the termination site called the terminator utilization substance. And I'm not going to lie, I can't help but seeing Arnold Schwarzenegger every time I say that, and thinking I'll be back about replication, which is true, right? Lots of replication, lots of times. So let's write this down. And note that replication is bidirectional. That's what I was showing, two different directions, until the replisome meets at the termination site on the other side. That's where the terminator utilization substance will be bound and will stop the process. It will cause those replisomes to leave the template DNA, and the process of replication will be complete. Now, we should recognize that a replisome is a very complex machine. It's composed of many proteins and many different kinds of proteins. So we'll zoom in on the replosome in a little bit and talk a little more about what's found in it. But suffice it to say that it's a large complex heading in these two directions. And here we see a time-lapse shot where now it has made some new DNA. The new DNA is what we're showing in red. So you can see what we call a replication bubble opening up, where each replosome has headed in different directions. And at each area where a replosome shoots around is that it's actually forming what we call a replication fork. Now, if you want to, this isn't labeled conspicuously in your notes, but this whole thing here is a replication bubble. So we can try to show that this is a replication bubble. Now, the replication bubble gets bigger and bigger and bigger as the replisomes head around around the existing DNA, using it as template. So you can see here that now the replication bubble has become quite large. So there's an extension of that bubble with time until finally over on the other side it actually meets that terminator utilization substance and the process stops. And now the replication bubble is so large as to be two separate strands of DNA. So it is now completely copied the genome of the cell. And it's interesting to note that in that process it is actually semi-conservative, because one strand, of course, was used as the template to make the new strand. So one strand is new and one strand is old. Now, seeing it as a pressed flat on the page sort of representation is really quite false. And in fact, we know that the DNA is a double-stranded helix. And so we will recognize on the next slide a little bit more accurate, perhaps, depiction of this replication process. Before I go there, though, what I want to note is that, in fact, it takes about 40 minutes for this process to take place. So despite the fact that the replisomes are wicked fast, they have about 3,500 base pairs to copy here, so they have quite a job on their hands. And so with that note, you might be thinking to yourself, and I don't know, like for some reason I'm picturing Caleb with the light going on and he's thinking, wait a minute, Rachel, that doesn't make any sense at all. Didn't you just say that this was E. coli? And haven't you said 1,000,000 times in lab that E. coli can double in like 20 minutes? So if it takes 40 minutes for it to replicate its chromosome, how in the world can it double in 40 minutes? That is ridiculous. And in fact, it is impossible. But what happens is, notice that here in this image, that in fact another round of replication could begin at the same time as this round of replication is going. And that's how that's accomplished. That's how the doubling time of a culture can actually be faster than the replication time of the chromosome. Crazy! Here's a picture showing a more accurate depiction where you can see the double-stranded helical nature of the chromosome. And we can see the forks a little better, and notice that the purple DNA is what's showing the newly synthesized DNA. Do note that in this image, the replisomes are not pictured, so they would be big complexes hanging out at the fork in each case. So let's zoom in and talk a little bit about what are these crazy complexes in the replosome. What constitutes a replosome? Well, the most important thing in the replosome is DNA polymerase. In fact, DNA polymerase 3 is the name of the enzyme that is actually doing a majority of the synthesis of DNA. So we're going to call it DNA pol 3. DNA pol 3 in prokaryotes is what does a majority of the making of DNA. And DNA pol 3, oh my goodness, it is incredible. Like, this is a wicked fast enzyme. Now, I'm talking about speeds of 750 to 1,000 base pairs per second, ramping along, heading along the template DNA, building new DNA. And of course, in order to build new DNA, it has to have building blocks. The building blocks, of course, to synthesize new DNA are the nucleotides. But we will refer to them by their more complex name and their more accurate name of deoxynucleoside triphosphates, or DNTPs. Now, this can get kind of a little bit confusing, because when we think about this, the D refers to the fact that this is DNA, right? Deoxyribonucleic acid. So the building blocks also have to be deoxy, meaning that that 2' hydroxyl has to not be present, right? It's not RNA, it's DNA, so that's not hydroxyl in DNA. Now the NTPs, that's just a generic way to say any of the various NTPs. So this could be an A because the base is adenine. It could be a G because the base is guanine. It could be a C because the base is cytosine. So we could have DTTP, DCTP, DATP, DGTP, right? These would be the more specific names for each of the deoxynucleotide triphosphates. Now let's draw it just so that everybody remembers what this looks like, recalling that a nucleotide, and this is just going to be kind of a cartoon depiction without all the details, but remember it's got a base here, bound to carbon number 1. That's a nitrogenous base, and it might be G, it might be A, it might be T, it might be C, right? And then attached on to carbon number 5, we have the phosphate groups. So in this case, in a DNTP, it is a triphosphate. So there are actually 3. I'm going to have to be a little creative here, fitting all 3 of these on. But there are 3 phosphoryl groups. And I'm not drawing all the chemistry on there, on this DNTP. So when the nucleotide itself gets added into an elongating chain of DNA, it will break this bond, this phosphoester bond, and we'll see the release, of course, of energy and the addition of a nucleotide onto the ever elongating chain. So we will recognize then that these DNTPs, and I should add in here the 3' hydroxyl, that is ever important because we know new nucleotides are added onto that open 3' hydroxyl. So this is the building spot for new DNA. And that's what I want to talk about now, because although DNA polymerase is incredibly fast, and it really is an amazing enzyme, it has some restrictions. One of those restrictions or needs is that it has to have building blocks to build with, so it needs to have DNTPs. But also another restriction is that it can only ever build if it has an open 3' hydroxyl. Without an open 3' hydroxyl, it has nothing to add onto. So I think of this as being kind of like some of those race cars that I know Sarah had mentioned she was kind of into cars, but Sarah Mark, right? So, this is like one of those race cars that you have to like push it in order to get it started. So this is kind of what I think of when I think of DNA polymerase. Wicked fast, but of course it has to have a jump start, and it needs to have that primer. So it needs to have an open 3' hydroxyl. We call that a primer because we know it needs to have a start before it can actually get going. So the other restriction is that it always builds in the same direction. So it's always building on that open 3' hydroxyl, which means it's always building in the 5' to 3' direction. So it needs a primer, always builds in the same direction, and has to have building blocks. These are the needs that DNA polymerase has. So although it's incredibly fast, it is also an incredibly needy enzyme. Holy cow, we know people that are a lot like that, right? Okay, so let's look at an example here where we have a 5' to 3' strand of DNA on the top. And on the bottom, we of course have the anti-parallel strand. We know that's always how DNA works, 3' to 5'. And we want to know what DNTP is going to get added next. That is, will it be DCTP? Will it be DGTP? Will it be DTTP? Or maybe is it DATP, right? Which one is added next? Okay, well, we have to think about the needs of DNA polymerase, right? Really needy. It's got to have an open 3' hydroxyl. So we know it's either going to have to add here or it's going to have to add here. Okay, good. All right, so we know that, and we know it will only build in the 5' to 3' direction, so it's only going to either build this direction or this direction. It would never build, so we could do this. It will not ever build that direction, right? I think I'm getting a little loopy tonight, but it will either build one of those 2 directions. But hang on, DNA polymerase is even more needy, because how would DNA polymerase know what to do if it didn't have a base to pair with? It has to, right? Because how do we know whether we incorporate C, G, T, or A? Well, we know A pairs with T, T pairs with A, G pairs with C pairs with G, so it's got to have something to pair with. So if we think about the idea of DNA polymerase adding to this 3' hydroxyl, how would it know what to pair with? Question mark. There's nothing there for it to pair with. However, if we look here, this is my kitty Cal, so I'm going to introduce you. Maybe. That's Cal, being obnoxious as usual. Sorry. So how would it know what to pair with? Well, if it added here, it wouldn't know, right? That's a big question mark. However, if it adds right here next to this open 3' hydroxyl, check it out. It's got a great pair right there. So let's erase a little bit of stuff here. And just note that right here, it would know that it would pair a T with an A. So there's our answer. The next DNTP to get added is DTTP. So let's go through and see if we can build a little bit of DNA, see how good of a job we can do at being DNA polymerase. Now I know for one thing is sure, we definitely are not as fast. So we go DTTP will be next. Oh look, another DTTP, DCTP, DCTP, right? This is how DNA polymerase would do it, only it would do it way faster than I can do it. So not only that, this is where I get really jealous because not only is it fast, but it also is incredibly accurate. And guess what? It gets to carry around its very own, get this, proofreader. It has its own proofreader that tells it, hey, you made a mistake, back up, do it again. Oh my gosh, what I wouldn't give to have a 3' to 5' exonuclease that is a proofreader. It causes the DNA polymerase holoenzyme to turn around and fix mistakes if it makes them. And yet it's doing this so rapidly. So this is has high fidelity. It is an incredibly awesome enzyme, and I wish I had a 3' to 5' exonuclease to come around and cut out my mistakes and fix them after me. Let's take a moment to zoom on into the replication fork, and I want to look at the replisome specifically and how it's building DNA as it opens up the first little tiny initial replication bubble. But before we zoom in, and this is going to be our zoom in down here, I want to make sure to bring up the fact that there are other components involved in the replication fork. So let's recognize that one of the things that has to happen is that the DNA has to be unwound. So there's a very awesome enzyme called helicase that unwinds DNA. And this, I believe this is the point where Eric really wanted to stand up and take the floor and talk about helicase. I don't know, maybe it was ligase you wanted to talk about, but I bet you wanted to talk about helicase as well, because the epic pickup line is, I wish I was DNA helicase so that I could unzip your genes. So DNA helicase does unwind the DNA. And of course, anybody who knits a little bit knows that in fact, if you kind of try to rapidly unwind yarn, it's going to all tangle. So DNA topoisomerases are around to relieve any of the tension that might take place in rapid unwinding of DNA and to take care of the issues with supercoiling ahead and behind the replication fork. So topoisomerase and helicase play a really important role in the process of replication. Even though obviously they're not the ones that are building the DNA, they play such a super important role there. So let's now follow the replisome and see what's happening actually at both replisomes. So I'm going to draw this in. This is a zoomed in view. So we could say, okay, here's one of our replisomes. And we know it's going to head that way. Here's another of our replisomes. And it's going to head that way. And within both of these, we're seeing synthesis of new DNA. So here we're seeing that this replisome here is making one strand like this that is synthesized anti-parallel to the existing strand. And it's making another strand like this that's anti-parallel to this strand. And we recognize that with the progress of this fork, that replication bubble gets larger and larger. But somebody out there is watching this and they're saying to themselves, whoa, Rachel, slow down, stop the press, because look at that. This strand of DNA was synthesized 5' to 3', adding new nucleotides to the 3' hydroxyl. That looks good. Happy days, right? However, this one, look at this, being synthesized 3' to 5', adding to the open 5' phosphate? Wait a minute. No. Heck no. Doesn't happen. Never happens. So this strand can't get synthesized that way. So we have a problem. This strand here, by the other replosome, 5' to 3', no problem. That can happen, right? That works out well. However, the other strand here, sorry about my splooge, the other strand here, this one cannot get synthesized 3' to 5'. So this is another one of those heck no's, right? Not going to happen. So while we see these two strands that we are going to term the leading strand getting synthesized comfortably in this direction, we're going to have to talk more about what happens with this lagging strand here. It can't get synthesized the way that I have shown. We'll go ahead and write some of that down and say synthesis is continuous in the direction of the fork for one strand called the leading strand. This one and this one. The other one, the lagging strand, well, that has to happen discontinuously. So I don't know who else likes to knit, but this is a lot like knitting actually, where you know how if you're knitting with multiple colors, you have one color that you just keep in one hand and you just grab it each time you knit, right? It's very continuous. The other one you keep in the other hand, and in order to get it integrated, you have to loop it back around with your other hand. So you go like this and loop it back around, and then you knit continuously with the one strand, one color, and then you loop the other one around. And continue. Okay, well this is exactly what DNA polymerase does. In fact, it has to loop back around this lagging strand in order to build it. It has to loop it back around so that it can synthesize it in the only direction that it knows how, in the 5' to 3' direction. So let's talk about the details and nuances of that process of synthesis of the lagging strand.